Hello everybody, my name is Lena Alnazer and today I'm going to be discussing my research. So my research was basically a method development for the detection of sulfated flavonoids in plant tissues. So to kind of begin and introduce my topic, we're going to first be talking about gypsum soil. So gypsum soil is a unique soil type which is composed of more than 16% of calcium sulfate dihydrate. And this soil has a high concentration of sulfur. And this high concentration of sulfur makes it difficult for the plants to live on it. So one kind of mechanism that can be possible for these plants to survive on the soil is to store the excess sulfur in sulfated flavonoids. So sulfated flavonoids are a pigment produced by some taxa in leaves and floral tissues. So we used a model species, was Leycinia californica, which we do know these do contain sulfated flavonoids, and we wanted to test a method to see if we can detect the presence or absence of these sulfated flavonoids. So if the method worked to actually detect the presence of sulfated flavonoids, then we could use this for other species that actually do live on gypsum. Another kind of goal for this experiment was to determine the amount of mass we could use in the analysis and the method development to figure out how much we could use for future species. So to talk about the methods, we grew our Leycinia californica in a greenhouse, and then we cut the flower heads, we dried them into an oven, and then we grounded them with using a Wiley mill. Then we varied different masses of the ground flower heads, and we had 3 replicants of each mass. We then added methanol to extract the flavonoids, and these test tubes sat for 24 hours. After this, TLC analysis was performed, and then ultraviolet light was shown to detect the presence or absence of a yellow band. So we had previous research done that we were trying to mimic and figure out we can compare to. So we have a TLC plate that is shown on the project by Dr. Nishi Rajakaruna, which basically the yellow band showed the presence of sulfated flavonoids. So with our data, we used around 20 milligrams of the ground flower heads, and we did see a presence of of the yellow band, which indicates the presence of sulfated flavonoids in Leycinia californica, which we did already know. So this showed the method did work. And we also did this with a smaller sample mass, which is 0.7 milligrams, and the band still was shown. So even with a small amount of mass, we could still see the presence of yellow bands. So this being said, the method did end up working for the detection of sulfated flavonoids, and we were able to evaluate that the greater the mass of the ground sulfated flavonoids, the more vivid and visible the yellow band was. If we compare the 2 bands on the poster, we could see that 20 milligrams was a lot more vivid than the 0.7 milligrams. However, they still were shown, which is great. So to move for future work and whatnot, we would like to use this method since we do know it works to detect the presence of sulfated flavonoids. We want to use this on those plants that live on gypsum soil. So if we do this method and we see the presence of yellow bands on the plants that live on gypsum soil, then we could evaluate and say that the plants that live on gypsum soil do use sulfated flavonoids as a mechanism to survive and as a way to store their excess sulfur. So that was the main basis of our experiment. We were able to have a successful method development, and thank you.